Right. Okay. Get going. Welcome. Hi, how are you? Hi, guys. Welcome. Welcome for joining me. So, I have a list of topic areas, but is there anything you guys want since you are the first in here that you'd like me to start with? Morning. Good morning. Good morning. Good morning. Hi, Eloise. Hi, everybody. So yeah, I've got a list of topics that people have put on from yesterday, but either if there's any topics you guys want me to start with, otherwise I'll just start with my list. Cardiac cycle, absolutely. Let's start there because I do feel like cardiac cycle is actually coming. I feel like how they're going to ask it may be linked to ECGs. So I would definitely say that that is a topic you want to ensure that you have covered. So what we'll do then is we'll start with the cardiac cycle and then we'll go from there. Everyone's happy with that? So it's an interesting one with the cardiac cycle because obviously you're talking about the fact that it's not just understanding the cycle, it's understanding the pressure changes in the heart, it's understanding how that links to neuronal communication as well, and how that also links to the part of where the hormones can affect the body. So you want to make sure that you are covering all of it interlinked together. So on this one, let me just do this. Transparent plants, what are they like? The things they actually know. Classification evolution, I'll actually do next week. I'll try and do another one of these next week for you because that's paper 2, classification evolution. So today I want to focus solely on the stuff which is to do with the cardiac cycle, that is to do with the way in which the body responds. So module 5, module 3, and module 2. Module 1 obviously is your process of application skills for any practical topic area. So do make sure that when you're doing this, that you are happy with that because they can bring it up in any which way they want. Perfect. Right. So obviously, if we were looking at, let's start here. If we're looking at the cardiac cycle then, so the first thing that you want to be able to do is to note that the cardiac cycle is literally either going to be asked where you've been given a diagram like this. So it's all about pressure changes. But what's really important when you're looking at the pressure changes in the cardiac cycle is that you understand that that is also linked to the way in which the heart actually beats. So if you understand that the heart is obviously made up of your different chambers, you should know you have atrial systole, you should know that you have ventricular systole, and you should know in the graphs where different things are going to close and where different things are going to open when we talk about the valves. So it's important to note when you are describing the cardiac cycle and you're describing the way in which the heart responds is to do with the pressure changes. So you can quite clearly see that my blue line, my left atrium, so the pressure in my left atrium is greater than the pressure in my yellow line, my left ventricle. So up until the point where the yellow line crosses the blue line, my atrial pressure is actually greater than the ventricular pressure. I haven't put a Zoom link in actually. No, what I can do, let me do that now. For those who want to come in, and then once it's filled, it's filled. There we go. So what you have is your Atrial pressure is greater than your ventricle pressure up until the point in which it is actually going to link together. So if you understand that your pressure changes in the graph. This is the bit that a lot of students tend to struggle with, in the fact that they think that they just need to say, oh, the atrioventricular valve is closed, or they need to say that the semilunar valve is open or the semilunar valve's closed. Atrioventricular valve is open. And you memorize that content. And then the question doesn't ask you about that. The question asks you for where you are actually looking at what is going on. So at what time did, for example, the atrioventricular valve close? That would be where they're assessing your module 1 content. So if they are asking you to analyze a graph about the cardiac cycle, then they are looking for you to note the increase and decrease. They're expecting you to note that they are both following the same pattern: increase, decrease. Then we have the increase here, but our aortic pressure is greater. At what point? So do make sure when you're going through these questions or these topic areas that you are looking at the time on your axes or the pressure on your y-axis. Yes, I will cover that as well as I'm going. So it is important that when you're looking at the cardiac cycle, you don't just memorize atrial systole, the atria are contracting, ventricular systole, the ventricles are contracting, diastole, the heart is relaxing. It's important that you're linking it all together. What does that actually mean? What does atrial systole mean? And what does ventricular systole and what does diastole mean? So make sure you know about the sinoatrial node. Make sure you know about the atrioventricular node. Make sure you know how to label if they were to ask you about these different structures. So it's a really interesting one, because when I did this before, I always talked about non-conducting tissue, preventing impulses moving directly to ventricles. Now, it's an interesting thing that the more I do this, the more I research, the more I learn new things myself, the more I add to what I am teaching, the more I want to improve that. And so therefore, I have now got a new bit I want to add in here. If you give me 1 second. I'm going to add in a new part to this. I'm just going to quickly find it. So many things open. Try and help you. So when we talk about the muscles, and when we talk about the histology, so someone's asked me about histology and going through histology. One of the things I do feel that is potentially going to come up is the difference between the cardiac, the smooth, and the skeletal muscles. So there's going to be a lot linking those together. And if that's the case, they're not just expecting you to understand that you've got your SAN node, your AVN, you've got your bundle of His per count fibers. They will expect you to understand that you've got your cardiac muscle that is striated. What does that mean? If it's striated, it's striped. Does it have branched fibers, or does it have the myofibrils, for example? Is it uninucleated? Is it multinucleated? So this is something I do feel like they could ask you. And so what you need to note is, when we talk about the cardiac cycle, we talk about the cardiac muscle, we talk about differentiated cells, module 2 coming in. What you have is this connections between the cells, which we call these intercalatal discs. Well, a lot of students will go, well, I've never been taught that. I don't need to know that. Well, you have been taught about cells, module 2. You have been taught about cell membranes, which are the biological membranes, module 2. You should know that what you have is the fact that these membrane proteins, membrane structures, phospholipid bilayer, which has these proteins in between, these connect those cells together, and because they connect those cells together, what that does is that forms a channel between the membranes, and what that allows is for them all to contract together as synchronized to allow for that atrial contraction. Contract, contract. So when we talk about atrial systole and we talk about ventricular systole, what we're actually talking about is all of the cells interconnected, contracting, all of the cells interconnecting, contracting. So they're going to contract not together, separately, one, then two. And because of that, we know when we look at the electrocardiogram that there is going to be key areas that show us P wave, atrial depolarization, QRS complex, ventricular depolarization, T wave, which is about ventricular repolarization. And you'll be able to see, oh, I'm not put it in this one. Hold on. See, this is where I'm using old PowerPoints and not changed it. Here you have the gap between the P and the QRS. This is the delay. So this is the delay that you have between your atria contracting and your ventricle contracting. So because you have a delay, so you have the contraction, 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 contraction. What we're showing here is that you need that slight delay. Otherwise, if we don't cause that delay to occur, then what you'll get is both of them contracting at the same time. And remember what I said before, it's about pressure. Pressure being able to force the blood up and out, because remember, it's going from the heart all the way around the body, up to my head, out to my fingertips, down to my toes, and back to the heart again, if we're looking at the systemic circulation. If I'm looking at the pulmonic circulation, it's going from the heart to the lungs and back to the heart. Not as far to go, and therefore it doesn't need as much of a force, but you still need to have those pressure changes. So it is important to note that you don't just memorize P, Q, R, S, T waves, that you actually look at what that means going forward. Yes, Naveen, I'm going to bring that one in. What's the slight increase? Okay, give me a second, Lois, and I will cover that bit. And then this was from the muscle topic part. Yes, so if you're talking about the branch cells and the fact that they allow the electrical impulse to spread, that's good. It's not about allowing cross bridges. Be careful you don't confuse the cross bridges when you talk about the muscles and you're talking about the fact that the actin and the myosin head go together, and that you don't get those wordings confused. That's what a lot of students do. This is why I'm pinpointing what you've said here. A lot of students memorize these key words and then they try and throw them in at the wrong points and they don't make sense. So if you just remove that cross bridge out, saying that the branch cells allow for the electrical impulses to spread through the atrial tissue, that is correct. And that will allow you to get that marking point. So the fact that it's branched allows for that simultaneous contraction. Yes, that's correct. What's the purpose of the delay? So Eloise, the purpose of the delay is to ensure that you have that contract, contract. We don't want it to contract at the same point, because if we're pushing at the same point, we're not going to get that all the way around and back again. Right, let me just double check these. Is there any access for the ECG? Like, what do the peaks actually represent? Yes, so if they give you it in the exam, they may show you that it's, for example, 1 decimeter cubed. Let me give you an example of that one. She says, if I can find one here. Let me show you a question. Where am I? Where am I? Where am I? Which one is this? Oh, it's because you're in the way. Let me move you out the way. Yes, this is the one I want to go to. So if we were looking at, for example, this question here, you can see on this question where they've said they've given you time, so 0.2 seconds. They've given you here some information about 0.2 millivolts. So we're talking about the potential difference. So when you do your nerves and you know about depolarization, repolarization, hyperpolarization, what we're seeing here is that this is showing us the electrical activities, the membrane potential between them. So I hope that's helpful. But if they're asking you a question, they will give you that information so that you can work it out. Sometimes I will use exam papers from different exam boards or questions that you haven't seen, because actually that helps you to work it out. So what about after the delay, after the QRs? So this is an interesting one, because obviously we've got going back. So Tony, that's a good question. If I now come back to here, so we have the QRS complex. So remember, that's the depolarization of the ventricle. And then what we have, so if we bring this back in, depolarization of the ventricle, we do have a slight delay before we have that repolarization. So remember that depolarization that's happening in the ventricle, we want the ventricle to contract from the apex up. And what it's going to do is it's going to squeeze the blood out. And when it squeezes the blood out, that will then force it to where it needs to go. So there is a slight delay so that we can make sure we're squeezing as much out before we then relax. Because the T wave, that ventricular repolarization, that is where you're going to have that diastole, that relaxation of the ventricles, as well as the atrial diastoles already occurred. You want it to be that they can now start filling up again. So it's about a delay between the P wave and the QRS complex, which shows you about the AVN. Now, somebody mentioned earlier about how the animal response links to this, and that links quite nicely to this point here. Let me just bring in this one. So if we were looking at here, paper 1, animal response, and we're looking at, we've got our SEM, we've got our AVN, we've got our baroreceptors and chemoreceptors. You should know that these are part of your animal response topic area. So the baroreceptor is obviously detecting the pressure. So they are mechanoreceptors. They're detecting the fact that the blood is being pumped out, and that's going to cause a slight increase in volume of the blood that's leaving. So the cardiac output is going to change. That's going to be detected by that receptor, the baroreceptor, which is a mechanoreceptor. So you will have learned about the mechanoreceptors. That's the name you need to know. They may in the exam give it as an example of the baroreceptors in the aortic arch detect and so forth. So make sure you know the term, the mechanoreceptor, because that's the word they're wanting. If you were to write the baroreceptor as an example, they will also allow that in the exam. So you've got the baroreceptor, you've got the chemoreceptor. So we're going to be looking at that pressure of the blood being pumped out. We're going to be looking at the pH, the chemoreceptors. So therefore, what that's going to do is that's specialized cells, receptors that are going to detect. They are transducers because they're going to convert that into an electrical impulse. So now we've got the sensory neuron, which is going to pass that information to our central nervous system. So from our receptor to the central nervous system. So in this case, we're at the medulla oblongata, because the medulla oblongata is where the coordination is going to happen for our autonomic nervous system. So in this case, what we have is our sympathetic nerve is going to be activated if, for example, I need to increase my heart rate. So if I've been doing lots of exercise, my heart is obviously going to need to pump faster. Well, my sinoatrial node is exciting itself. And because it's exciting itself, it's got a rhythm in which it's going to activate. So I want to speed that frequency in which it sends the depolarization across the atria. So to increase that, I'm going to have a message in electrical impulse go down my sympathetic nerve. And when it gets to the synaptic bulb, it is then going to release neurotransmitters, these chemical messengers that are going to bind to receptors on the sinoatrial node. So if I'm going back, because someone did mention about hormonal system, if I'm going back to the hormonal system side of things, what are we talking about here? We're talking about hormones being released, or are we talking about animal response, neurotransmitters being released? They are chemicals which need to signal for something else to happen. So whether they are neurotransmitters that are binding to receptors on the surface of the membrane, or whether they are the non-polar hormones that are getting through into the cell, depends on what they're looking at. So in this case, we're looking at neurotransmitters, those neurotransmitters binding to the receptors that are at the sinoatrial node. Therefore, they are spreading that depolarization across all of the atria, causing the frequency of the depolarization to occur at a faster rate. So when we talk about sympathetic stimulation, we're increasing the heartbeat, increasing the frequency, and it is really important that we talk about those words, the increase in the frequency of the impulse. If we have a really strong stimulus, so for example, if I have gone to my classroom, this used to happen all the time, and somebody would come in and they had literally doused themselves in perfume or deodorant and they'd walk past me, what would happen is it was a strong stimulus and I would get a really strong sense of the smell, because the frequency in which my neurons were passing that information on was a lot faster, because there was a strong stimulus. If there's a low stimulus, then a low frequency of those impulses will be sent. Tony, I'll come back to that. No, you don't have to answer your question, located, you don't have to state where they're located. They will probably give that to you. That's not an expectation. They expect you to know the different types: photoreceptor, mechanoreceptor, chemoreceptor, and thermoreceptor. The only one that you have to know specifically, because it's named in your exam, will be the Pacinian corpuscle. But the others, they could give you completely random examples. They expect you to know those key ones, so you should be able to work out whether it is a photo, it's to do with light, whether it is a chemo, to do with chemicals, so ions, or whether it is a pressure or movement. So they have given random examples where it was to do the statholith moving around. So that was a mechanoreceptor because it was to do with movement. So yeah, they'll give you that. What hormones cause the heart rate to decline? Do you need to know? So you don't need to know about the hormones that are going to decrease. You do need to know about the hormones that are increasing, but you need to know about the parasympathetic nervous system, which will slow it down. So just to finish what I was saying here, when we look at the heart rate, you're looking at the shortening of the delay. And this is why I wanted to talk this through. When we talk about the shortening of the delay between the sinoatrial node and the atrioventricular node, what happens is the atrioventricular node actually slows the impulse down before it passes it on. And the reason being is we want that pressure building up so that we can squeeze the blood out. So if we've got the sympathetic stimulation, if we're increasing the frequency of the sinoatrial node, we also want to increase the speed at which the AVN is going to pass on that information. So therefore, we want to shorten the delay, that gap between the P wave and the QRS complex. So if we have our sympathetic nervous system in play, there would be a much shorter delay, a much smaller gap between that P wave and the QRS complex. Obviously, we're going to increase the force of contraction, which will then affect your cardiac output. So your cardiac output is your heart rate times your stroke volume. So do make sure, if they're going to ask a question on this, you've checked the numbers. Is it decimeters cubed or is it centimeters cubed? Is the heart rate in minutes or is the heart rate in seconds? So your beats per minute should be in the heartbeats per minute. So you may have to do some calculations there. And Tony, I will come back to that in a second. And increase blood flow supply to the heart because coronary arteries need their oxygen and glucose for respiration. So they could bring in respiration, but link it to the animal response and link it to the questions which are relating to that contraction and the cardiac cycle. Give me a second. I'm just going to look at the questions now on, sorry guys, if I'm missing your questions on YouTube, I will come. I'm having a quick read. I will cover hormonal responses. Hormonal response regarding the heart rate and control would be adrenaline. Yes, it's all about ensuring that you're fully emptying and allowing for the filling up to get that pressure to get the blood out. Yes, that's correct, Phoebe. That the T wave is about the ventricular repolarization, which occurs during diastole. How do you know the heart question that was just up, like the heart rate? Yeah, so that's the question. Hold on, let me go back because Tony also asked that. I did say I would refer back to this. So what happens during diastole, and to answer Tony's question as well, diastole is about relaxation. And that's where, if you think about your muscles, and I will bring in muscles here in a minute, when we talk about muscles, we talk about muscles relaxing because this is a striated muscle, right? The cardiac cycle is a striated muscle. It's a striped muscle, and therefore it needs to contract and relax. And it does it in a way in which we've already learned about striated muscles, but more linked to skeletal muscle. So in the fact that it's contracting and relaxing, what we're talking about is the fact that the sarcomeres are shortening, and when we're relaxing, the sarcomeres are going back to the original position. Yes, amna repolarization has to occur during diastole, because you've got atrial systole and then atrial diastole is happening whilst ventricular systole is occurring. But we don't tend to talk about it because we're so focused on the ventricular systole at that point. But you do have the atrial diastole occurring at the same time as ventricular systole. So, Sahib, to answer your question, is it not the accelerator nerve? There, you can call it the accelerator nerve. You can call it the sympathetic nerve because what I'm showing you is that it's the vagus nerve and it's the accelerator nerve. But it depends how your teachers taught it to you. They're not going to penalize you if you just say it's the sympathetic neuron and don't name it because it's important that you know the difference. There is no expectation that you know accelerator nerve and vagus nerve. There is an expectation you know that it's a sympathetic nervous system and the sympathetic nervous system. And that's what they're looking for. So no, Naveen, you wouldn't say the receptor on the right atrium. You would say the receptor on the sinoatrial node. It's really important to say it's on the sinoatrial node, not the right atrium, because the right atrium is very big. And it's only activated by the sinoatrial node. So that's the important part. Do we have to mention noradrenaline as the hormone when sympathetic nervous system is activated? So be careful. It's an interesting one. With the sympathetic nervous system, noradrenaline is the neurotransmitter. Noradrenaline is also the hormone that can be released by the medulla in the adrenal gland. So noradrenaline can be a neurotransmitter and a hormone because they are chemicals which are released either through the release from the neuron, it's a neurotransmitter, or from the adrenal gland, and therefore it's the hormone. But basically, they are chemical messengers. So you won't be able to see either of them because they are too small. You wouldn't have the So the question Amna's asked is, would you be able to see the sinoatrial node and atrioventricular node? No, you wouldn't unless you were looking at the magnification and resolution being really high because they're so small, so you wouldn't see them, no. So the valves opening, when the valves are opening, it's all to do with pressure. So if I go back to my diagram here, a second. So if we go back here, when you're looking at the part here, you can see that the valve is shut. So if the valve is shut, that's telling you that the pressure below is greater than the pressure above and greater than the pressure below. And that's why you have that difference. It's all to do with the pressure. So here my blue line is greater than my yellow line. So the pressure at this point here, the pressure above up here is greater than the pressure below. And that's because the blood is filling and it's passively going to flow into the ventricle. So Beatrice, to answer your question, how does it act as a neurotransmitter? Noradrenaline is a neurotransmitter if it is released by the sympathetic neuron. So if it's released from a neuron, it's a neurotransmitter. If it's released from the endocrine gland, it's a hormone. What's the difference between adrenaline and noradrenaline? It's just the structure of them. They don't expect you to know the difference in the structure, but they do need to expect you to know that both of them are released from the adrenal gland and from the medulla of the adrenal gland. So what you have to understand is you've got the, so to answer your question, when it comes to the frequency of the impulse from the sinoatrial node, so the information has come from the accelerator nerve. There has been a neurotransmitter release. That neurotransmitter has bound to the sinoatrial node. The sinoatrial node is going to send the depolarization through the atria so that the atria contract at the same time. So what we're saying is the information has been sent down the sympathetic nerve to the sinoatrial node. The sinoatrial node is then depolarizing. So the atria are contracting, and then that frequency is going to increase again. So what we're going to get is the sinoatrial node are now sending the depolarization across at a faster rate. So what you're getting is contraction, 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 contraction. Whereas, if I go back a second, oh, no, I want to go here. If we're looking at this one, what you can see is the delay between the T wave and the P wave, the delay between the T wave and the P wave, and the delay between the T wave and the P wave. You can see a difference. So you can see that frequency changing. So if I have got, for example, I'm doing some exercise, what I would expect is that my trace would be normal. I'd see a PQRS and a T wave, but I'd see a shorter delay between the T and the P wave because the heart is pumping at a faster rate. So the frequency at which the depolarization is going through the atria is faster. So the difference, Tony, to answer, what tips did you give her? I'll kind of drip them in fizz it all the way through as I'm doing it. Have a dip down. Okay, so with regards to the ectopic pregnancy, ectopic heartbeat, apologies. So when it comes to an ectopic heartbeat, what we're talking about here is an extra heartbeat that's occurred, and therefore you don't get the normal trace with an ectopic heartbeat. And so therefore that's why you don't see the PQRST wave exactly the same. But the difference between the ectopic and the fibrillation, you can see here very carefully. If you look, there's my PQRST, PQRST. Then I've got, I can't really see my P wave. So my P wave is not really there. There's my Q and there's my T. But now I've got this dropping down section here. So that means there has been an extra heartbeat without that delay. And that therefore is where there's a problem. So if you have a extra heartbeat that shouldn't happen, you get this confusing diagram. So that's what they would expect. With the atrial fibrillation, you cannot tell where the P and the T waves are. There's no distinct P waves, but the QRS complex should be pretty normal. But if we can't distinguish where the P and the T wave are, what we're saying is the atria are going through fibrillation because they're contracting quite quickly. And so that would be where they're distinguishing between the difference. And then just to finish off, going back here, because somebody did ask about how can you tell? With this question here, they told me that this was 0.2 seconds. So 10 little boxes was 0.2 seconds. So I would go from a P wave to a P wave. So in this case, I have 23 boxes. That's one heartbeat. So 23 boxes. And I know if there's 10 boxes that equal 0.2, I know each one is 0.02. So 23 times 0.02 equals 0.46 for one heartbeat. So then I would take 60 divided by the 0.02. So that would be what I would say if you're asking on that one. Okay, I want to move away from the cardiac cycle and I want to bring in muscles because I do feel like that will be quite a big topic area. Let me just bring this one quickly across. So when, Zach, to answer your question, what must you remember to include when drawing your ECG is to ensure that you have your P wave, your QRS, and your T wave. Make sure that you, if you've been given a diagram above, to copy and to then, let's say they ask you to draw atrial fibrillation, not atrial fibrillation. They have done that in the past. If they ask you to do atrial fibrillation, do not make it obvious where the P and the T waves are, but make sure your QRS complex is the same. That's what they're looking for. If they're asking you for a tachycardia trace, then look at whatever they've given you as a normal, copy exactly the proportions, but just make the T and the P wave delay shorter would be what I would suggest. If we're talking about decreasing the heart rate, you're absolutely right. The parasympathetic nerve is stimulated and the acetylcholine is released. That's absolutely right. So that again, acetylcholine now binds to the receptor on the sinoatrial node, and therefore we're going to decrease the frequency of the depolarization. That's right. Acetylcholine is not always excitatory, but if they're asking solely about neuronal communication, then you talk about it being excitatory, unless they've given you some hints in the question. Absolutely correct. Yes, the P wave is atrial systole. Yes, Abira, I am going to bring in plant transport, and I feel like it's more translocation on that front, and. Why isn't acetylcholine always excitatory? Well, it depends on the channel. The acetylcholine is the neurotransmitter that's going to be released. It's about which channel is it attached to. So if the acetylcholine is going to bind to a receptor and that receptor is on, let's say, a potassium channel, if that opens the potassium channel, potassium is going to flood out and therefore make the membrane hyperpolarized. I don't know, Zach, to be honest. I'm just going to go. I've got a meeting at 12:30, so I'll see how long I can go for, but I'll try and give you guys as much as I can. And I will obviously try and get as much into this, but I'm going with just whatever and whoever is here. So yeah, I'm just going to keep going with it. Elle, I am going to do histology. I'm just about to bring it in. Don't think I'm not seeing these messages. Don't keep bombarding me with the same messages though, because it'll just make me not want to do it. Sorry, I came late. The other thing I would say is if you are joining me late, guys, this is on my YouTube channel, so it is recorded. So you can go back and watch this again and you have points to it. Okay, so one of the things, if they are going to ask you about the histology and they're looking again at muscle. So remember I said cardiac muscle, I said smooth muscle, and I've also said skeletal muscle. So if they're going to give you a histology of it, they are expecting you to note that the involuntary muscle is not striated. They're expecting you to note that it is spindle shaped, so it's not branched, and it's uninucleated. So if I were to make an assumption of a question they may ask to compare them, is for you to say, for example, smooth muscle are non-striated, whereas skeletal and cardiac are both striated. Smooth muscle are spindle shaped and uninucleated, as is the cardiac muscle, whereas the skeletal muscle is multinucleated. So if they're going to assess that content and it be a nice question, it will literally just be those bits they're looking for. When we talk about smooth muscles or involuntary muscles, that's the same thing, by the way. We talk about slow contractions, long contraction length. So, for example, peristalsis, if I'm moving the food down my esophagus or I'm moving food through my intestines, I want slow contractions, and the contraction length is going to be long, and that's because I'm going to decrease, because we're contracting here, decrease the space. If we're thinking about our intestines, decreasing the space so that my enzymes that have been produced are going to be able to collide more successfully with substrate so that they can break them down. So that's why we've got slow contractions, but long contractions, because as well, we're moving things through our intestines. Let's go to 12:30. We'll see. Skeletal muscle, striated muscle. Why would we want slow contractions in the trachea? Well, it's an interesting one because when we talk about contractions, the trachea, you want to stay open, okay? It's more thinking about the smooth muscles, Abira, in your bronchioles. So if I've got, for example, a response to, I have taken in a allergen and it is causing a problem, I'm going to want to constrict my airways, or I might want to dilate my airways. So I don't want it to be a rapid contraction, because if it's a rapid contraction, then it's going to run out of the ATP molecules a lot faster than if it's a slower contraction. So it's a completely different type of muscle, and the way in which it does the contraction is different. The only one you have to know about with the sliding filament theory is the skeletal muscle. Understanding that it's a slower contraction allows for that constriction of the airways or the dilation of the airways. Does that help? Good. Okay. So just going back to skeletal muscles, striated, fibers are tubular, rapid contraction, because the skeletal muscle is under somatic control. I am thinking about the fact that I'm moving my arms right now. Actually, half the time I'm not, and it's just who I am. But I am using my conscious somatic nervous system. And so therefore, I want these rapid contractions for me to be able to contract, for example, my bicep or contract my tricep, because they work antagonistically. So uni means one nucleus. Multi means many nuclei within the same cell. So if you have, for example, one nucleus, we know the nucleus is where our genetic material is contained. Our genetic material in our chromosomes have therefore got our genes which are going to be transcribed and translated, and therefore producing the proteins which are required. Multinucleated, many nuclei, means we've got much more genetic material within the cell. Therefore, we can actually repair, for example, those proteins that might get damaged through the process of overexertion. Yes, it does. So actually, let me just bring in a quick one on that in that question you asked. Ba, 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 ba. So you should know the autonomic nervous system because this is still module one, paper one. That's what I meant. Parasympathetic nervous system and sympathetic nervous system work antagonistically. So inhibits heart rate, accelerates heart rate, constricts bronchi, relaxes bronchi. Obviously, the sympathetic nervous system is going to activate adrenaline and noradrenaline from being released. The sympathetic nervous system stimulates glucose release from the liver. So this is where you've got your hepatocytes store glycogen by polysaccharide. By polysaccharide then will need to be hydrolyzed down. That hydrolysis happening will release the glucose molecules so that the glucose molecules can then be put back into the bloodstream so that they can circulate round to the muscles and then stimulating your digestive system, stimulating your gallbladder, and inhibiting digestive activity. So they work antagonistically. And because they work antagonistically, it means that sometimes you'll have one accelerating, one releasing, and one not releasing. So one inhibited from releasing, or it may be that they're both releasing, and it's about how that then affects the signal that's coming through to the postsynaptic neuron. So remember that one of the things that someone actually mentioned earlier was about summation. Summation is the adding up of neurotransmitters, and therefore, if you're adding up the neurotransmitters effects, so if I've got a stimulatory, so I've got one that depolarizes the postsynaptic membrane, I've got one that inhibits the postsynaptic membrane by causing hyperpolarization. If they've both been released at the same time, that's going to keep the postsynaptic neuron at the resting potential. So actually what you have is lots of different neurons releasing neurotransmitters, and it's the summation, the adding up of them, which causes either the impulse to be sent on or not. Yes, so if we're talking about the smooth muscles contractor, yes. So if we're releasing the neurotransmitter, so okay, give me 1 second actually, because it links quite nicely to this. There are two muscles. You don't need to know this for OCRA, but it annoys me they don't teach it to you because then it becomes confusing. If I were to look inside my iris, for example, so you know pupils constrict and pupils dilate, okay? So what you should know, or you shouldn't know this, but it helps you to know this, is there are muscles which are called longitudinal muscles, which go all the way around, okay? And there are muscles which are called radial muscles that do this. So if I was looking at still in my circle, my radial muscles. So they're going to cause the contraction potentially, but that contraction will be either to shorten and therefore dilate the pupil, or if the contraction is going to be the longitudinal muscles, that's going to therefore cause the constriction. So basically what I'm saying is when we dilate our pupils, we are activating our radial muscles, and when we are constricting it, we're actually activating our longitudinal muscles. So what you have to understand, those lines are representing the muscles that are going to contract, and you have two different types. Those that are longitudinal and those that are radial, and it's about which one's being activated with which one is going to actually cause the constriction. So that therefore means that you did, Zach, triple, whereas if you did combined science, you don't need to know this, but that's how it works. And it's the same within the esophagus and in your intestines. It's to do with these different muscles. What you need to know for this is whether it's the parasympathetic or whether it's the sympathetic. Okay, so yeah, they removed blinking. It used to be the cranial reflex was in there, but they've only gone on knee jerk. They removed it for some reason. A muscle contraction experiment. Um, Muscle contraction wouldn't really be an experiment if they were looking at, I don't know, to be honest. I do feel like there's going to be a lot on animal response, but because animal response can bring in so many different topic areas, because it can bring in neuronal communication, it can bring in your muscles, it can bring in the transport in animals, it can bring in cells, it can bring in the liver, it can, so it basically is all interlinked. So I do feel like that it's all going to come together as one. Um, the knee-jerk reflex is actually quite simple in the fact that all they want you to know is that with the knee-jerk reflex, it's few synapses because it's a reflex, few synapses involved, and the only thing about it is you're going to stimulate one muscle to contract while you're going to stimulate another muscle to relax. So you're inhibiting or you are stimulating the muscles to do what needs to happen. Give me 1 second. Let me just throw in a quick bit about this. Where am I going? Animal response. And then I will cover muscle, and I am going to do plants. What time are we on? 10:30. A question, Christine, where have you put it? Where have I put it? Where have I put it? Where have I put it? Because I've updated it. Oh, I think I just saw it. Reflex action. There we go. Here we go. This is where I've got too many things and I keep updating and updating and changing and because I want to make sure that you guys have got the best possible stuff. So I'm always improving. Right, reflex reaction. If they're talking about a reflex action, what are they going to ask you? The key thing they're going to ask you is, whatever it is, do you understand that because it's a reflex action, it's extremely fast, so few synapses involved? That's like a key go-to. If we're talking about reflex, we need this to be fast. We need to prevent danger, but we don't need to consciously think about it. So when I made my girls some cupcakes this morning. When I was getting it out of the oven, I was lifting something that was really hot. So therefore, if I didn't have a towel there, if I went to go and grab that hot bit of metal, what's going to happen is my thermoreceptors on my fingers are going to detect the temperature. They are therefore going to send the impulse up to my central nervous system because that is what is going to coordinate. My central nervous system is going to coordinate. That's going to therefore need to come down to my effector through my motor neuron, and that needs me to let go as quick as possible. It needs for me to release and not actually burn myself. So therefore, I don't need to involve my synapse because if I did, it would go receptors, sensory neuron, really, 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 really motor, and then the effector and release. So we don't want that. That's a too long a period of time, which could damage the cells. They wouldn't be able to function. That puts us in a disadvantage. So therefore, the evolution that occurred, this is the best possible advantageous process. So it's not that there's no relay neurons. I'm going to show you, Eden, in a second. It's the fact that there can be no relay neuron and a relay neuron, in the fact that normally we would say stimulus receptor sensory relay motor effector synapse. So what we've got is the shortest pathway possible to get a response that keeps us able to function. So we are doing this without any conscious thought, therefore it is involuntary. Yes, it's part of the autonomic nervous system because remember, we're talking about the autonomic nervous system. The autonomic nervous system is our peripheral nervous system. Peripheral nervous system is your sensory and your motor neurons. The central nervous system is your brain and spinal cord. No, it's not. Be careful. Your reflex, and this is why it was bad that they removed the blinking reflex. The reflex is the extremely fast response involving few synapses. So when you look at, for example, the blinking reflex, it's called a cranial reflex because you've got a receptor in your eye, a sensory neuron, then you've got your central nervous system, the brain, to the motor neuron back for you to blink your muscle, the effector, which causes you to blink, you're closing your eyes. So it is not just about a spinal cord. It also could be to do with the brain because remember, the medulla oblongata is part of the brain and the medulla oblongata is going to be controlling, coordinating those autonomic nervous responses. Addy, I haven't forgot your question. I am going to come to that later. And normal that there is just one. Yes, a few synapses, Tony. So if we look at the knee jerk reflex, see, I'm going blah, 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 too fast. And actually, I would say to you, if you do that in the exam, sometimes if you're like me, where your brain is on basically autopilot, extremely quickly moving, what can happen is you can write the wrong thing. So I would say, try if you are in that exam and you know you do things a bit too quickly, make silly mistakes, try and take a breath back, slow yourself back down again. Easy for me to say that because this is what I'm doing. I'm a but I'm not sitting the exam, but I know that in the exam I will speed up too quickly and I will make some silly mistakes. So last year, after my student sat the exam and I got access to the paper, I sat and did the paper as if I was a student and I made some silly mistakes. And I made some silly mistakes because I wasn't taking the time to read the information and also I wasn't breathing because I was getting stressed by it. So one thing I would say to you is try your best to take some time to just breathe. If I get access to it, I will potentially try the paper again. I do every year because it's quite good for me to put myself into the place of where the students are, but I won't have access to it the same way that I did last year. And so I'm relying on all of you, to be honest, to come out of that exam. I don't care about you asking me about what the answer is. What I care about is what topic areas came up, what were the questions like, so that I can gauge where paper 2 and paper 3 may go. So yeah, but I will try it, but I won't be able to access it in the same. They don't upload it for me to access it until normally I would say about a week and a half after you've sat the exam, which doesn't help you. Anyway, back to knee-jerk reflex. This one is a spinal reflex. So this one specifically is a spinal reflex because what you have here is you have a sensory neuron, and that sensory neuron is being activated because there is a receptor. So when you hit at the patella here, what's going to happen is that is going to affect a mechanoreceptor that will detect the stimulus. That will then be a transducer in the fact that that will convert that movement to that pressure change, the hitting of the patella, that will change that into an electrical impulse. So that impulse is now going to be passed by the sensory neuron down to the spinal cord. So this diagram here is showing you my spinal cord. So the fact that I have my spinal cord, why is it a mechanoreceptor? It's a mechanoreceptor because it's not light, so it's not a photoreceptor. It's not temperature, so it's not a thermoreceptor. It's not the other one. My brain's now gone. So light, temperature. It's not an osmoreceptor, so it's not that one. So chemoreceptor. That's the one I couldn't think of. It's not a chemoreceptor, so therefore we're talking about pressure or movement. So this has to be a mechanoreceptor. So in my joints there are mechanoreceptors. So that's how I knew it was a mechanoreceptor, because it was to do with the movement, so pressure and movement. And then what you have is the mechanoreceptor detects it, it's a transducer, it's going to convert it into a generator potential. That therefore means that my sensory neuron is going to be transferring the action potential down to the relay neuron. So this one here, this green one, is showing you a relay neuron, whereas the blue one here, so you can see this one here, it's connecting straight to a motor neuron. So the action potential would be, are you saying how's the action potential transferred between the mechanoreceptor to the sensory neuron, Abira? Is that your question? So, well, what have we got between? If we looked here, if we were to, hold on, give me a second, zoom in. Gonna zoom right in. What have I got? I've got a gap. So I have a synapse there. So therefore, I have a postsynaptic neuron and a presynaptic neuron. So therefore, I have a neurotransmitter that's going to be released from my presynaptic neuron, the sensory neuron, to my postsynaptic neuron, the relay neuron. Or if we're looking at this one here, then what have I got? I've got my presynaptic neuron, which is my sensory neuron, and my postsynaptic neuron, which is my motor neuron. So it really just depends which one we're talking about, but we're talking about a neuron. A neuron, we've reached a synapse, so it has to be a neurotransmitter. So the action potential is propagated down the sensory neuron. The neurotransmitter is released. It's going to diffuse across the synapse, and then the postsynaptic neuron is then going to pass that information on. So then you have, if it's not present, what does it tell you? There's some sort of problems with the nervous system, multiple oscillations where it kicks out lots of time. Could be that there's a cerebellar disease, and that's because your cerebellum is the one which is going to coordinate your balance. So this is a way in which they could detect that. And that could be how they assess your knowledge here, by linking the knee-jerk reflex with the brain structure. And do you know that the cerebellum is for coordinating your balance? Yeah, you've got two different neurons. So basically you've got you've got one which is a inhibitory and one which is a stimulatory. So we don't want both muscles to contract at the same time. We want it to kick out and then come back in. So therefore, for it to be able to kick out, we want to stimulate the muscle at the top, but we want to inhibit the muscle at the bottom. So it's about the fact that muscles work antagonistically, and that's why you would need both of them there. Right, I'm going to quickly spin on with the muscles, because otherwise we could spend loads of time here, and I'm very aware of the time, and I want to cover transport implants as well. So just to quickly go over the muscles and sort of questions they could give you. If they give you an electron micrograph to do with muscles, make sure you know what a muscle cell is. So a muscle cell, we call a muscle fiber. A muscle fiber is made up of myofibrils. Myofibrils are cylindrical organelles. Ooh, cell structure, module 2, cylindrical organelles made up of proteins. So these are really long organelles which are made of these proteins, and these proteins are surrounded by sarcoplasmic reticulum, which have calcium in them. What you should know is, if you look up here, each one of these, the V is showing you one of those myofibrils. So there's a myofibril, there's a myofibril, there's a myofibril. So then what you need to note is if they ask you a question, can you actually label where X is? So I'm just going to quickly run through these. These are things that I did in my course that I ran the last couple of weeks. So sarcolemma, it's a plasma membrane. That's not correct. Sarcoplasmic reticulum releases calcium ions. Sarcomere would be X, so that would be the right answer. And the myofibril will be each of these red circles. So this would be the whole thing across here. So make sure you know what you are labeling if they give you a question like this. You should know the thick filament. You should know the fact that it's got actin binding site, ATP binding site, hinge. You should know thin filament, troponin. You should know tropomyosin. You should know that it's blocking cross bridges, that it is moved out of the way when you are contracting. Do you need to know about the H zone, the I band, and the A band? No, you don't need to know them, but you do need to understand the dark band and the light band and the striation of it. So you should know your Z lines, you should know your M line, you should know that each of these is a sarcomere, and a sarcomere is a contractile unit. The reason you have what's known as the light band and the dark band is because the thin filament is attached to the Z line, the thick filament is attached to the M line. And therefore, when we look at this, there's my Z line, there's my M line, therefore my X to Y is a sarcomere. So what would T be? Now, this is a really bad picture, but T would be showing you a mitochondrion, or it could be a glycogen granule. In this one, I believe it was actually named as a glycogen granule because they actually wanted to know, did you understand that that glycogen granule would be broken down into glucose to provide the necessary respiratory substrates for respiration to occur? So how would you know? And this is why I'm saying it could possibly, because it depends on the picture that they show you. This one was a glycogen granule. How would you know? It would either be like this if it's a long shape, or you would expect it where you could see it looking like that. So it would be very obvious in the question which one it is if they want you to say that it was the mitochondrion. So it's important to note when they ask those types of questions, they will do it based on clear diagrams, or they'll give you a little bit of information that tells you, for example, it was a source of glucose, or it was a necessary for respiration, for example. Sorry guys, I keep forgetting you're over here. Sorry, I've just seen that operant condition is not part of your OCRA, so no. Masood, what are you struggling with the chemoreceptor heart rate bit? Yeah, so my myofibril is made up of many sarcomeres. How do you tell if it is a nucleus or a granule? The nucleus would be larger, so we'd expect a larger nucleus if that was the case on that front. Okay, so if we're looking then, the light band, which is the I. Now, for those of you who are like, I've learned about the I and the H, and because you can see I've got different diagrams that I've stolen here and used. So these diagrams show you with regards to your H zone, your I band, your A band. How do I remember the different ones? I know that light has got the letter I in it. Sounds ridiculous, but that is how I remember it. Light is the I band, it's actin only, whereas the dark band has got an A in it, it's the A band. That's not why it's called that, just to be clear, but that's how I remember which way round it is. So the I band is my light band, and therefore it's actin only. My A band is my dark band, and therefore it's myosin with some overlap. Now your H zone is the area where there is no overlap. So it's important to know the H zone is basically, if they're asking about that, where my M line is. That's what they're really asking you if they're asking you the question. So you should know that your sarcomere is going to be shortened. Yes, it will, Zara. That's why I'm doing it here. It's on, I'm live streaming, so which means it will save it straight onto my YouTube channel so you can watch it later. And today's the day I can offer a bit of my time because I've got clients all day tomorrow and Wednesday, and I've got something on Thursday. A band where myosin actin overlap or the length of myosin. So with regards to the A band, I would be just talking about the A band being the dark band. I would be saying that it's the myosin because it depends how much of the overlap there is. The dark band never changes. Abira, I won't potentially be live on Wednesday. I will go live Wednesday night as usual, but I've got clients all day. Wednesday's like my big, I teach 7 hours on Wednesday. So I teach all day, but I will go live on Wednesday night after I've done that. I just don't have time during the day. Whereas today is normally my planning day, but I've given up planning to do this for you because I know everyone's feeling a bit tense. Why does the A band not change during the contraction? Right. Okay. This is an interesting one. It's a good question, Addie. So what you have is, I'm going to use my arms and my, it's a bit of a weird one, but if you imagine that my face here is my M line and my elbows are my Z line. I don't know if I can do this properly, but so Z line to Z line. So out here, my M line is here. So basically what we're going to say is that my Z lines are going to move closer. You can see, ignore my hands, but I have to do this. You can see that my elbows are getting closer to my M line, okay? Because you can see my elbows are getting closer to my M line, that means my Z lines are getting closer to my M lines. My sarcomere is shortening, okay? Now, ignore that. So I'm not going to show this part. I'm going to now do it with my arms, okay? So if I put my sleeves down. So now my face is my M line and my black shirt right up to my elbow here. This is the myosin. So therefore, that doesn't change shape. So the myosin stays like this. It's basically like the M line and the myosin are always like this, whereas my Z lines can move. So therefore, the dark band never changes. So hold on. This is where I try and do an analogy and it doesn't actually sometimes make sense. Give me a second. I'll come back to this. So what I was saying is basically, there I am with my head. So my head is my M line coming all the way down. And with my black arms, with my shirt that's black, that's the dark band. So that goes all the way down. That's the dark band. Right? That's the dark band. Dark band. So now I've now got my light part of me. So my light part is my arms without my sleeves, yeah? So now what I'm doing is I'm going to imagine, I'm going to draw a different, let me get rid of, go away. 1 second. Get rid of that. So now what I have is this part and this part, that is my, from my elbow to my hand, from my elbow to my hand. So my arms don't change, but when I look at it this way, that can come towards, so the light band gets shorter because we're being pulled towards the M line. So when you look at it, yes, yes, it is. So if I go back to, oh, hold on a second, I've lost you. Where are you? Where are you? Here we go. If I go back to here, what you can see is my sarcomere has shortened because remember it's moving in both directions. My light band has changed and got shorter, but my dark band hasn't. My dark band stays the same. If I go back, look at the dark band, it doesn't change. The dark band stays the same. Classification evolution is not in paper 1. Classification evolution is paper 2. I will go live or before paper 2 and paper 3, depending on when I don't have clients. I've got a lot of clients who I'm seeing just before their exam. So when I can, I will do it. Module 4 is paper 2. It's module 3, module 5, and 1 and 2 for paper 1. So here you have the sarcomere gets shorter, dark band doesn't change shape, and we know this because it's sliding filament theory. That's all right. Okay, so if you're given a diagram that looks like this, straight away, what you should be able to see is I can see my dark band. I can see my dark band, I can see my light band, and therefore, because I can see my dark band and my light band, that therefore means I can work out where my Z lines are and where my M line is. And if I just rub out this here, you can quite clearly see where the H zone is, if that helps. Right. Okay, so therefore you're looking for how to label each one of these. And let's just look at this one. So neuromuscular junction. So they are definitely going to ask questions. I feel like I'm shouting at you all. Sorry if I'm getting louder. What's I? Hold on. Going back here. I was the myosin or the thick filament because you can't see, so you can expect the fact that you're in the dark band that it's myosin. Okay. Why isn't it myosin and actin? So going back to here, how can you tell where the actin is overlapping? So if I have my elbows like this, it's really hard to do. So if I have it like this, right? So here's my dark part and my hands, and I'm going to use my fingers as my Z line. Where's there an overlap? So where's the dark and the light overlapping? Is it all the way up to my head, or is it only up to halfway through? You can't see the overlap in this picture. So therefore, that's why you wouldn't say it's myosin and actin. You would just say the one thing you know for sure, which is myosin. Zara, we're about to do muscles and I am going to do transport as well. So I need to move on. But basically, absolutely, paper one stuff. Okay. The circles in the picture are the glycogen granules. Yes. In this one, yes, I do believe so. Okay. Acetylcholine is released because we're looking at a neuromuscular junction. I know that's part of my somatic nervous system. So I know it's acetylcholine. I know that they're going to diffuse. And the other thing to remember about this is the fact that calcium coming in is going to cause the vesicles to fuse. So you need to bring in knowledge on. So let me just add in here. The fact that I've got diffusion, what they could do is expect you to note the difference of, well, if it's diffusing in, it has to be a high to low. We're going across a phospholipid bilayer. Lipid bilayer. And because we're going across a phospholipid bilayer, therefore, we need a protein. Where did the protein come from? Well, the protein is obviously made through the process of transcription translation, and therefore the information for that calcium channel has to be within the gene. When the cell differentiated, so this is where they could bring a lot of the topic areas together. We've got cell differentiation because we have a motor neuron, it's a differentiated cell. We know we're looking at a membrane, so therefore we've got the phospholipid bilayer. We know we need calcium to get in to cause the exocytosis of the neurotransmitter, so exocytosis to occur. So therefore we know that we had to have the cytoskeleton involved because the cytoskeleton was what moved the vesicles towards the membrane. Why are they able to fuse together? Well, we have a membrane-bound organelle. We have a plasma membrane. They are both the same. So because they are made of the same structure, a phospholipid bilayer, it means that they're able to fuse together and they're also able to pinch off. Potentially, I don't think that they would ask you to compare the cholinergic synapse with a neuromuscular junction, but they could. And if they did ask you to compare the two of them together, then you're looking at the fact that they're both releasing neurotransmitters, the fact that The cholinergic synapse and the neuromuscular junction both release acetylcholine. However, the fact that one of them is going to, be careful, be careful. It's important to note that you are stating that they are both depolarizing the membrane. So the sarcolemma and the postsynaptic membrane being depolarized is due with sodium channels, so that would make it the same. However, saying that the depolarization links to T tubules and then causes the release of calcium ions rather than the depolarization causes a propagation of an action potential. I don't feel like there's enough to compare the two, but it doesn't mean they couldn't do it. But similarities, absolutely, there's loads of similarities that you could talk about. Also, we call the calcium. Yes, you really do need to call that protein is a voltage gated calcium channel. That is very important. No, Zara, you don't need to compare the action. The only action they'll expect you to do is acetylcholine unless they've given you information. However, they do expect you to know what noradrenaline would cause. So if it were released, what would it cause? So if it's part of, obviously, noradrenaline is a parasympathetic nervous system. No, it's not. That's an absolute lie. Noradrenaline is the sympathetic nervous system, and therefore, because it's the sympathetic nervous system, its activity, and therefore the speeding up of the heart rate, the dilation of the pupils, etc., the dilation of the bronchi, the intercostal muscles. They could link it to the intercostal muscles in the diaphragm and the changing of the volume, increasing the frequency of the breaths. I'm not sure if they will do that, but they could. The reason there's no action potential in the neuromuscular junction is because it's a different cell which has a different set of channels and a different role to play. So the reason we just talk about the depolarization of the membrane is because that depolarization, the increase in the sodium ions, sodium flooding in, causing the membrane potential to change, is going to activate the sarcoplasmic reticulum to therefore open its channels, these voltage gated calcium channels, so that they will then release the calcium ions into the sarcoplasm. So the reason we don't call it an action potential is because it's not a depolarization, repolarization, hyperpolarization. It is a voltage gated calcium channel opening and calcium being released. Whereas when we talk about an action potential, it's those three things. Depolarization, the sodium, voltage gated sodium channels open. Repolarization, the voltage gated potassium channels open. Hyperpolarization, where we overshoot the resting potential. Whereas in this case, when we're looking at the action of acetylcholine at the neuromuscular junction, we're talking about the releasing of the calcium so that the calcium can bind to the troponin, therefore change its shape, therefore move the tropomyosin out the way, therefore we have the actin and the myosin form cross bridges. So we're talking about the depolarization. So the membrane is depolarized. And because the membrane becomes depolarized, that's where we get the voltage gated calcium channels opening. So then if we're looking at, yes. So be careful, the difference between depolarization is the opening of the let's try that again. Depolarization is the opening of the sodium channels so that sodium floods in and therefore depolarizes the membrane. Depolarizing of the membrane is where the membrane comes away from the membrane potential. whatever that resting state was. So when we talk about, for example, the release, and someone did ask me to do this, when we talk about the hormonal system and the releasing of the insulin, the membrane becomes depolarized because the potassium ions can't leave. So if we're talking about depolarizing the membrane, we're talking about taking the membrane away from its resting state. When we're talking about depolarization, we talk about the sodium channels opening and sodium flooding in. So it's really important you get the difference between what depolarizing the membrane means or hyperpolarizing the membrane and what depolarization, when we talk about neuronal communication, and repolarization. No, 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 Amna, don't get yourself confused on that. The membrane is depolarized, which then activates the voltage-gated calcium channels to open. Calcium is now going to leave through the channels out into the sarcoplasm. So the generator potential, generator potential is the potential, you changing the potential difference. So yes, we would talk about the transducer is the cell, and what it does is a transducer because it's going to turn a stimulus into a generator potential. The generator potential is where we have reached the threshold value and therefore we have generated the potential. Because the acetylcholine that was released from the motor neuron was bound to receptors in the sodium channel. So the receptors open the channel because the acetylcholine binds to it. Sodium floods in, depolarizing the membrane. Remember, your sodium potassium pump is going to be pumping sodium and potassium in different directions. Sodium is going to be pumped out, 3 sodium out, 2 potassium in. And because of that, what you're then going to get a concentration difference. So when acetylcholine binds to the receptor, that's therefore going to cause that channel to open, sodium can flood in. The voltage gated calcium channels are going to be activated to open when they have reached the potential difference. So when that depolarization of the membrane occurs. And that's why it's a form of positive feedback, because the depolarizing of the membrane is activating more channels to open. If we're looking at the sodium potassium pump, excuse me, and the sodium coming in and then the voltage gated sodium channels opening, sodium floods in. Whereas when we're looking at the calcium channels open, the calcium's flooding out. That's going the opposite way. Yeah, because we're pumping the sodium out, right? So the extracellular fluid is going to be made up of ions. And that's why the body has to maintain, and excess salts are excreted by the kidneys because we need to keep the balance the right way. Why does the sodium flood out? Because why does the calcium flood out? Because the calcium was being stored in the sarcoplasmic reticulum. The neuromuscular junction, the release of that acetylcholine, that is a chemical messenger to signal for the muscle to contract, for the calcium to be released, so that calcium can now bind to the troponin. And when it binds the troponin, it changes its shape. Therefore, that's going to move the tropomyosin out the way and therefore allow for the cross bridges to occur. Okay, good. Yeah, Amna, you good? Perfect. Right. Yes, so I'm coming back to the question with the myosin head. So what you can see, if I go back a second, when I had the troponin with the calcium attached, the tropomyosin moved out the way, my cross bridges can now form. So just be aware that ATP binding site has got adenosine diphosphate and inorganic phosphate. That movement, that tilting of the head that happens, that is actually caused because of the ADP and inorganic phosphate being released. So someone asked earlier about what is the use of phosphocreatine. Phosphocreatine is basically the storage molecule, which is a storage of an inorganic phosphate to quickly convert it back to ATP because ATP cannot be stored. So what happens is when we have got this moving towards the M line, the power stroke, myosin head is going to pivot. The actin's going to move towards the M line. We are shortening the sarcomere. What we have done is the power stroke. ATP is going to bind, and that's going to break the cross bridges. When the muscle is actually relaxed, the muscle is relaxed because the ATP allowed for the breaking of the cross bridges. Now, if we have the contraction occurring, what we have is the calcium still bound to the troponin. The troponin is still changing its shape. The tropomyosin is still out of its way. And therefore, that ATP can now be hydrolyzed. And when it's hydrolyzed down to ADP and inorganic phosphate, it returns to its original position. And if it's returned to its original position, let me just see if I can bring in, there we go, a picture. So before, what we had is ignore the fact this isn't, oh, hold on. Give me a second, let me, annoyingly. Oh. See if this will work. There we go. All right. So we actually moved. There we go. Right. So we moved it to that position there, kind of. And I moved it to that position. And let me send this to the back. There we go. And I'm going to move that down here. Okay. Oh, crumbs. 1 second, guys. Sorry. There we go. So we broke the cross bridges. And when we break the cross bridges, the fact that it hydrolyzes, it therefore can now reattach. Look at the change in position. So what you basically have is it doing this. So it's constantly moving them towards by shortening the sarcomere. And what you have is lots of these myosin heads, which are basically attach, pivot, detach, and they're doing this, moving the Z lines towards the M line, and that therefore will shorten that sarcomere. And so that's the important part. So the tropomyosin has to be moved out the way for the muscle to contract. If the tropomyosin gets put back into position, that's therefore going to block the cross bridges from reforming. And that's how you actually relax your muscle. So ATP breaks the cross bridges, and therefore if the tropomyosin can get back in the position, it will block that contraction from occurring. So as I said before, the creatine, so the creatine phosphate is there because it's a storage molecule. It maintains that supply in the initial contraction that needs to occur, that quick contraction. Okay, I want to jump away from this right now, and I want to bring in, because I've done nothing on plants, and I feel like plants are coming in, and I'm very aware of the timing. So let me bring in a bit on plants. Listen. Okay, so plants and how things move. Where does the creatine phosphate come from? The creatine phosphate is in the cells, in the muscle cells, and it's a way of a quick access to ATP because that transfer substrate level phosphorylation can occur, the movement of the phosphate to make ATP. Um, Mary, I'm not going to do a live tomorrow, I'm afraid. I am basically trying to do as much as I can for you guys today because I have my clients all day tomorrow and Wednesday. So basically, I have to prioritize my income, obviously. So I'm really sorry. I'll try and bring in Eden some stuff on the practical side of things. They did a lot on transpiration last year. So, and they do bring transpiration in quite often. So I feel like, I don't know why, but I've got a feeling that they could bring in translocation, linking it to the storage in seeds and then germination linking to your hormones for plant hormones. That's where I would say potentially they will come in, but it really just depends. Okay, so you should be able to identify whether you're looking at the root, whether you're looking at the stem, or whether you're looking at the leaf when it comes to your xylem and your phloem. And you should know the difference between those. What they could do is they could ask you, for example, to do a calibration of the graticule. They've done that the last couple of years though, so I'm not entirely sure if they will do that. However, they could. But they could do anything, to be honest. There's a one thing I have my gut feelings about this, but I can't guarantee you what it's going to be. This is just my gut feeling. It's my predictions, but my predictions could be completely wrong. So it's better that you know all of it and you can then hopefully link it together to whatever they ask you. The foundations of biology are the same. It's about applying the concepts to the situation. What are we looking at here? We're looking at differentiated cells, which are now grouped together with a specific function to play. Therefore, we are looking at tissue. Those tissue come together to form an organ. What is that organ? The vascular bundle. So if they're looking at assessing you on cell structure, they'll be looking at, did you know it was a cell? And did you know what happened to that cell? If they're looking at differentiation, do you know that groups of cells coming together have got a specific role to play, and what is that role? So when we look at the differentiated cells in the xylem, they are dead. They have got rid of all their organelles. They've got rid of everything, but they lignified things before they got rid of those structures because they're a cell. They were a cell that were produced through the process of mitosis. So therefore, they started out with everything. And then as they differentiated, they got rid of the things they didn't require. And that's why we've got a hollow tube, because it is a dead cell. It does not have any genetic material. It does not have anything in between because that will stop the movement of the substances. So therefore, if they ask that, that's assessing your knowledge of differentiation. If they're looking at this, they're assessing your knowledge of, do you know how to graticule works? Do you know how to work out the size of something? Could you draw this? So I do believe a biological drawing is coming up of what? Who knows? And therefore, can you draw? Do you know what to draw? Do you know what's expected? They're not expecting you to do minute detail. Whenever you're asked about biological drawings, they're expecting you to make it large enough, proportionable. Do you have a scale bar? Do you have continuous lines? Are they labeled? Is there a title? They are the skills that we're assessing you on. So therefore, that's what you need to do. They're not asking you to be the artist at all. Just be aware of that. So do not stress about, oh, it doesn't look amazing. As long as it looks similar to what is in front of you, that's what they want. So basically, if you have got a eyepiece graticule and you are told that you need to measure, for example, the vascular bundle in this case, you can see by measuring the vascular bundle, I've got 13 divisions. Somewhere in the question, they will have told you what each division is worth. 25 micrometers, for example, in this question. So therefore, I literally just do 13 times 25. They may have told you in the question that you have got, hold on, this is where me trying to do maths on the spot doesn't work. So let me just quickly work out something. So if I have, so if I were told that 725 micrometers were 29 divisions on my eyepiece graticule, they would therefore, they've told me that, they've told me 29, they therefore want me to do 725 divided by 29 to get to my 25 micrometers equals 1, and then times that by the 13. So whatever question they're giving you, make sure you know what it means if they ask you. I just made that up, Amna. I made it up on the spot because sometimes I throw in random numbers and I'm like, ah. So I literally just did 29 times 25 to give me a number so I could work backwards from it. So in the stem of the question, they would tell you so many divisions, in this case, 29 divisions equals 725 micrometers. Use that to figure out the size of the vascular bundle. So you're working back in the opposite way. Okay, or they could give it to you where, so this was me looking down a microscope and I was looking at the root tip and I was therefore figuring out what is happening. And so I know that each of my divisions is 25. So because of that, I know that I could do 25 micrometers. However, I had to zoom in my picture because here they were 25, but now I'm zooming in. Again, if this is the case, they will give you the information, excuse me, in the exam. This is where I'm talking too fast. I'm taking in too much air. And so what we've got is our high power magnification, 400 times. They've told me that in the question, and they've also potentially told me each eyepiece unit is 2.5 micrometers. So that will be in the stem of the question. And then all they want you to do is the same thing. Do your measurements. Diameter of the nucleus is 4.5 divisions. Therefore, 4.5 times 2.5, it will be 11.25. So any type of question that they give you, you must take the time to read the information that they've given to work that out. Okay, so what have we got here? We've got a root tip that's going to go through mitotic division. So if I've got a root tip that's going through mitotic division, why did it go through mitotic division? Send it back. I'm going to go a bit off cuff here because I feel like this is needed. Why did it go through mitotic division? Well, we know we're at the root. We're therefore at the root meristem, and therefore we have cells which are undifferentiated. So this is where they could go in the question, in the fact that the root meristem has undifferentiated cells. So how is it being signaled to go through mitotic division? Well, this is where you need to understand that you have your different hormones. So you should know that you have auxin that's produced at the root meristem, and that auxin, along with cytokinins, let's help if I spell this right. Say that again. Are going to cause growth within the root. So auxin causes cell elongation and cytokinins causes mitosis. Be careful, be careful. It's an interesting one with regards to whether it's going to stimulate or whether it's going to inhibit, depends on the concentration. And this is an important one because if the concentration is low at the shoot, it is going to stimulate the growth, but if it is high at the shoot, it's going to inhibit it. It's the opposite way round at the root. So it's not about it doing one or the other, and that's an important part of this. It's about the concentration and how that concentration is going to have an effect on whether it is inhibiting or whether it is promoting, so stimulating the growth. So that's the key things. Guys, I'm going to pause the live just now because I need to nip to the bathroom and then I will be back. So I'm going to pause for a second. I will come back and finish this off. So if you are with me, stay with me, go have a break yourself, but I will come back. Okay, I'm back. So coming back to what we were talking about, it can be an inhibitor, but it can also be a promoter. So if we were looking at, and this is where people memorize things, if I was looking at roots and I was looking at geotropism, what you should know is high concentrations at the root are going to inhibit the growth. But you've got to remember that the cells, for it to be able to bend, the cells up here need to be able to grow and they need to be able to do elongation. So because they need to be able to grow and they need to do elongation, that therefore means that you are going to have a low concentration. So a low concentration at the top, a high concentration at the bottom, and therefore it's going to inhibit the growth at the bottom. So inhibit the cell elongation at the bottom, but promote cell elongation at the top. That will cause the bending of the root to go downwards. And that's where students fall short in the exam. They have memorized, it promotes it here and inhibits it here, and then they've given you a question that's looking at concentrations, and that therefore is like, oh, hold on a minute, that doesn't match up with what I believed. And so it's really important you know it's all about that concentration that's going to have an effect. So the cytokinin is to do with cell division, so mitotic division. So if you think about cloning, and I know this is paper too, but if you think about when you're cloning and you are doing micropropagation, you are going to put the cutting in rooting powder if you're doing the cutting, whereas if you're doing micropropagation, you're going to take the explant and place that into a petri dish, and in that petri dish you are going to have auxin in the agar plate as well, or the agar nutrient, as well as cytokinin. Cytokinin is there for the cell division, mitotic division, and the auxin is for the root. My brain's starting to slow down. The root growth. And so therefore you need both them together. Now, what I will say to you is, for those of you who are with me here, I haven't planned this, okay? I am not planning this right now. I am literally going off the questions that you asked, and that's how I work, that's how I teach. And so I want you to understand, you'll notice I got stuck there in my brain. Something happened to me, and I started talking about cuttings. And I actually didn't mean to talk about cuttings, but that's where my brain went. But that was a trigger for me to be able to come back to micropropagation. And for some of you, that will happen in the exam as well. When you're answering a question, your brain goes to, oh, I remember this. And you start talking about something that is irrelevant and not the question that you mean to talk about. So be aware. I've been teaching this for 20 years. Now, I've taught all the different courses. I've taught AQA, I've taught IB. I only came back to OCR and taught OCR 2.5 years ago because I started back at a new school, and I came in to help them and support them, and I hadn't taught it for a while. And so therefore, I had to do this and I had to learn it in a fast way. I am human just like you are. I am someone who teaches this and does this on a regular basis, but I can still make mistakes. And that is okay. There is not an expectation that you are going to get 100% out of 100%. So it's okay to make mistakes, but what you need to do is you need to reread your answers. That's the thing that frustrates me the most. The number of students who don't go back and reread their answers and check them, because actually, a lot of the time, you'll have written one word that you didn't mean to write, where you just got it twisted in your head. You were thinking something else and you wrote the wrong word. And that completely negates all this lovely writing that you've done because of that mistake. Please take the time to reread, and also be aware that it's okay if it's kind of going off in different areas, if it's helping triggering your thought process to get you back to right point. Okay, where are we at? Oh, 11:30. Wow, this has been going. So for those of you who just joined me, this is a 2 hour session. It's now going on a bit longer. It is being recorded. So it is being recorded. So therefore, you can go back and look at the things. If you put science incorrect, yes, you will lose marking points. We can't give you the marks if you're giving wrong science. So this is the other thing that students do wrong, in that they think, I'm just going to write everything and anything that I can in the answer. Some of it, if it's irrelevant, we ignore it. So there's a difference between it being irrelevant to being incorrect. Incorrect science, we will mark you down for. We won't completely, you won't lose all the marks, but you will lose marks by putting incorrect science there. So do make sure you reread your answer. It's an interesting one with plant hormones, because with plant hormones, they are really looking at, do you understand that hormones, whether we're talking about animal hormones, whether we're talking about plant hormones, hormones are chemical messengers which are going to bind to receptors, and they are then going to signal for something to happen within that cell. So if we were looking at, let me bring in what happens with cell elongation. I don't want that one. Here we go. I want to bring this one in just because I started on cell elongation. So if we're talking about auxin and what auxin actually does, the key thing for us to note is that auxin actually alters gene expression. So this is where plant hormones and gene expression could come in together in paper 3. So auxins alters gene expression to promote the cell growth. So what's going to happen? Well, what we have is we have our membrane. So this is module 2, our membrane, where we're going to increase the number of proton pumps inside it. What's going to happen then is hydrogen ions can then be pumped from the cytoplasm into the cell wall. So if the cytoplasm has got hydrogen ions, and those hydrogen ions can then move into the cell wall, what's going to happen is we have changed the pH in the cell wall. We also have the fact that we have these enzymes called expansins, and if they're going to ask a question, they will give you that information. So those hydrogen ions, hold on one second, let me think this through. expansins, enzymes, module 2, enzymes that are sensitive to pH, module 2. Yes, I did wonder where you disappeared there, Tony. So the enzymes, enzymes which are expansins, expansins which are sensitive to pH, that's module 2. Module 2, where we are also looking at biological molecules. This is cellulose. Cellulose is made up of polysaccharide because it's made up of beta glucose, which has 180-degree inversion, and also forms cross links between microfibers. So what we have therefore is that these expansins are going to change their shape, be activated, and therefore break the crosslinks. If they are breaking the crosslinks, that's therefore going to increase the turgor pressure, pushing the cell wall outwards. Therefore, the cell is actually taking in water by osmosis because there's now a flexibility of the cell wall that allows it to take in more water. The cell has expanded. That is what we call cell elongation. So what we're actually looking at here, topic-wise, is module 2. Cell structure, biological molecules, cell membrane, movement of substances into the cell, allowing for it to grow, enzymes. This is all interlinked together in one topic area, potentially. And that's what you have to do. You have to see the bigger picture. You have to see how it's all interlinked. Okay, moving on. So that's the bit about that. I want to talk about, let me bring in. Now, obviously, I'm doing a massive whistle stop cafe here of the topic area as much as I can, because these are the sorts of things that I do in my group sessions and in my courses. But obviously, I know not everyone can afford it, and I know that some of you are getting really stressed as you get towards the end point. But just know, whatever you get at the end of these exams, they do not define you. They are a starting point. I never finished my equivalent of an A level. I didn't, okay? And yet I sit here with not only an undergrad degree in biology, I have 2 postgrads and a master's from Cambridge University. So I know that you are stressing. I know that things are hard, and I know that this is going to be a challenging couple of weeks for you, but please do note, this is just your starting point. And for some of you, if it doesn't work out, it's okay to retake an exam. I had to retake my genetics exam at university 3 times. I could not get it, and I almost failed my whole degree because of that. It's okay. It's what you put into it. So I just want to put that message out there because it is something I know that a lot of you are feeling stressed. A lot of you are worried and you're cramming right now, which is why I'm offering this. But do note, you can actually still achieve whatever you want to do. Okay. They may give you, now this question actually came in one of the exams where they gave them a insert, and the insert was obviously the xylem, and they were expecting you to be able to do magnification. Be aware certain things. If it's the xylem, what are you looking for? You're looking for the lignified rings, you're looking for your bordered pits, you know that it's a hollow tube, and the xylem is made up of dead cells. If we're looking at the phloem, the key thing for us to know is our companion cells. The other thing to know is when we talk about photosynthetic tissue, they did so much on photosynthesis last year. It will come in, but I think it'll be very small, or it'll be multiple choice questions if they're looking at photosynthesis, unless they're doing a practical. Someone asked about practicals. I feel like they could ask you to plan a practical which would measure the rate of photosynthesis looking at a limiting factor. So that could be where they go on that one. Photosynthetic tissue, obviously, we know they are going to be things like the palisade cell because that's one of the named ones for module 2. And therefore, because it's a named measure, it's a named cell, it's important that you know it is going to be lots of chloroplasts, long, closely packed together, thin walls, large vacuole to increase turgor pressure. So 1 second, let me come back. I've got like 3 questions. How did I do that? Because I took it, I went to college, just to answer your question, Eden. I left at the end of year 12 and I got a higher. In Scotland, you can do a higher and then advanced higher. I got a higher in biology. I left school. I worked at Tesco for a year. I then went to college to do a higher national certificate in leisure management. I loved the biology of it, not the leisure management side of it. That was boring. So I did that and then I left again. I didn't do the diploma and then I waited a couple of years and I didn't go till I was 20 because it wasn't right for me. I wasn't in the right headspace. I now know it's because I've got ADHD and I didn't know that at the time. And my brain made me believe that I wasn't good at science and I wasn't good at learning and I couldn't retain the information. But what I didn't realize, it was about figuring out how my brain works so that I could then figure out how to get better. And that's basically what I did my master's on your belief in yourself and knowing that going into that exam is going to be challenging, but that's okay. Back to this. When it comes to questions like this, how are you going to be able to figure this out? Well, first of all, how would you measure the rate of photosynthesis? That's a good question. How would you measure the rate of photosynthesis? Well, we know that photosynthesis is where carbon dioxide and water are required, and in the process of the light dependent reaction, light is needed to be absorbed, photons of light, we are going to get photolysis occurring. Photolysis is where we're going to split that water down to replace the electrons that need to go back to photosystem 2, and therefore oxygen is a waste product. So one of the ways I might measure the rate of photosynthesis could be through the production of oxygen. And if I am doing that, I'm measuring the light dependent reaction because photolysis occurs as a waste product of the light dependent reaction. Now, I could measure the rate of photosynthesis by looking at carbon dioxide concentration. And if I'm looking at carbon dioxide and how much that's decreasing, I'm therefore looking at the Kelvin cycle. And therefore, I am looking at that fixation. So I would want to use, if I'm doing this in the right way, if I'm measuring oxygen, I'm going to be using a very basic way, you may have done this, counting the bubbles. So if I'm counting the bubbles, how big are the bubbles? That's not very effective way of measuring. So therefore, I would potentially want to use the volume, a gas syringe. That's a better way of improving this investigation. Rather than counting bubbles, I'm going to measure the volume. But hold on 1 second. Again, can I go a step further? Can I improve it? If I've measured the volume, can I improve after using a analog piece of equipment, another piece of equipment? I would therefore use potentially a oxygen probe. So absolutely right. It is all dependent on context of questions, whether we're talking about how many decimal places, how many significant figures. It depends on the question. It depends on the information that you're given. If you're looking at significant figures, you should be going to the significant figures of what the question has given you and the decimal points to what the table of results is giving you. You may go to one significant figure more in process data. So if you're doing process data, you can go to one significant figure more than what was given in the question. It therefore all depends on the information that you've been given in the exam. Yes, you would bring in statistical testing and everything. I'll come back to that in a second. So Amna, to answer your question, how do you measure carbon dioxide concentration? You measure carbon dioxide concentration using a carbon dioxide probe. What they could do is they could say to you, well, hold on a second, what about using an indicator? Because we know that carbon dioxide, when it dissolves in water, is going to dissociate. So we've got our carbonic acid. Our carbonic acid can then dissociate into hydrogen ions and hydrogen carbonate ions. And therefore, that can make an indicator solution acidic. That could be a way in which we can use qualitative data to assess whether something is actually happening. And so you can use a indicator to look at the carbon dioxide concentration based on how acidic the solution is. So therefore, we could use a pH probe. So there's lots of different ways. And again, it's all down to context of what the question gives you. And it depends what they want to do. If they're throwing in a random example, like the DC PIP experiment, a lot of students freaked out the Hills reaction because they'd never heard of DC PIP. No, it's not a mark a minute. It's 1.33 minutes per mark. Abira, to answer your question on that one. So yeah, so it's all about looking at what have they given you? What do you know? What can you relate it to? That's the important part. Percentages, ideally, percentages, you want to go up to a full percent unless they have given you specific information on that one. Okay, I will answer about statistical test on which one to go. Okay. Yes, I'm literally going on it now. This is where I'm going with this whole point of what I'm talking about. Because this is where I feel like they're going to go. Okay, Mariam, I agree that if they're going to look at photosynthesis, they could look at photosynthesis as it's happening at the source, or that it's a practical side of things, or they could look at the fact that photosynthesis, photosynthetic tissue, is going to be photosynthesizing to produce the glucose, i.e., the triose phosphate. And then the triose phosphate could be used to make the glucose. It could be used to make fructose, and those glucose and fructose molecules could then be used for translocation. So the movement of, so loading and the unloading and the assimilation of the glucose. So remember that you've got loading of assimilates, which could be sucrose and amino acids. Where did those sucrose and amino acids come from? The sucrose and amino acids came from the triose phosphate, which was part of the Calvin cycle, and you require enzymes for the conversions and the condensation reactions to join them together. Where do we get the amino acids from? Well, we need the amine groups. Therefore, we need the nitrate ion. So it's all interlinking. Yeah, that's how I teach, Mariam. I interlink the topics together. So you should know that that's what they'll do in the question. They're expecting you to know all of this. So the question will either be direct in that it's asking you about just your recall, or, and that's the bit that students struggle with, the application where it's interlinking it together. So you've got your source, your photosynthetic tissue, your leaves and your stems, chlorophyll molecules, TLC, they could check that. They are looking at you understanding that you have these accessory pigments, chlorophyll, a primary pigment absorbing sunlight energy, producing the products, reduced NADP and ATP, which are needed to fix the carbon dioxide so that we can make the triose phosphate. The triose phosphate can then be used to either make our sucrose or make our amino acids or make our oils, plant oils. And I will try and bring in lipids if I can. If I can, I'll try and do a quick video on it later if I have time. So the key thing is from the source to the sink, which could be the roots, the Murray stem cells, again, back to our undifferentiated cells. So how do they move things across? Symplast pathway continually through the cytoplasm, whether we're talking about the roots and we're talking about transpiration or whether we're talking about translocation, the Symplast pathway is about continual cytoplasm connected by plasmodesmata. Oh, interesting question. Yes, kind of actually. Explain. No, there's still application question. When you get an explain question, an explain question is can you recall and then apply it to the question in front of you? So it's a kind of joint apply and explain together. Um, so with regards to the Symplast pathway, continual through the cytoplasm, that basically just means that we have had photosynthesis occurring. The photosynthesis occurred producing our TP. Our TP is then going to leave the cycle, one of them, and because that TP leaves the cycle, it can then be converted through the use of enzymes within the cell. So that's the easy one. Literally, it's going to go from a high concentration to a low concentration. The apoplast pathway is different. This is where they'll bring in biological membranes and talk about transportation. Now, we know more about that because there's been more, and it's more interesting, more studies done on this. And what you know is your plasma membrane. Remember, plasma membrane, biological barrier. So therefore, we have a phospholipid bilayer that is not going to allow large, large disaccharides across that hydrophobic region. And so therefore, you require the transporters. Now, molecules can move across the phospholipid bilayer, but very slowly. And that's because it's fluid. Those phospholipids can move around. Because they can move around, like my hands moving around, that's going to create some space. And therefore, that's going to make moving of molecules through at a slower rate. So by having a protein inserted into the membrane, that's going to increase that movement, allowing these molecules across. Why can water move through the phospholipid without channels? Because, like I said before, the water can move but at a slow rate because of that phospholipids moving around. It therefore means that it can kind of quickly get through. And water is a small molecule. Because of that, we're talking about a larger molecule here. It would be even slower and therefore wouldn't be able to cross the phospholipid bilayer. But most of the membranes, biological membranes, are partially permeable because they have those aquaporins there. They have protein channels which allow water to move across. Abira, to answer your question, and it was one that Elle asked earlier, how does temperature and ethanol affect the phospholipid bilayer? Temperature, you know that temperature, straight away, any question that says temperature, kinetic energy. If I've got an increase in temperature, I've got more kinetic energy. More kinetic energy, therefore, means my phospholipids are moving more. They're like this, moving a lot faster. Well, if they're moving faster, they're going to create more spaces between them. So if I therefore have an increase in my temperature, I've got an increase in kinetic energy, more movement of my phospholipids, therefore a faster rate in which the substances can move because they've also got more kinetic energy. So therefore there's more leakage within the membrane. And the ethanol is a non-polar molecule. So what it can do is it can insert itself in between the phospholipids. And therefore it's basically like you and your best mate getting together and you've got a little sister that kind of gets in the way or a little brother that gets in the way and they're really annoying and they're kind of making it harder for you to stay together. That's what pushes you away. So ethanol has a part of it, the hydroxyl group, which is polar because of the hydroxyl group, but the other part is non-polar, so it can insert itself at the head and have a bit which disrupts the membrane. That's what you have when you have the ethanol. So if you've got more ethanol, you've got more disruption to the membrane. Do we mention hydrogenizing? Yes, we do. I'm just about to get to that in a minute. So Amina, does that answer your question? It has a polar part to it because of the hydroxyl group, but it also has a non-polar part to it. So it inserts itself into the membrane. Good. Okay, so if we're looking at the sucrose transporter, so we actually remove the sucrose here, and it then makes its way through the cell wall and into some intercellular space. Now what we need to do is we need to load it back into the companion cell. So what we do is we have the hydrogen ions being pumped. They're getting pumped against the concentration gradient. So it's active transport. So therefore we need ATP. So we would expect the companion cell to have lots of mitochondria for aerobic respiration. We have that being pumped across. We therefore have that it will come back in down its concentration gradient, and because it's coming back down its concentration gradient, that is going to therefore move the sucrose in with it through that co-transportation. No, it's for any type of movement of substances. So the apoplast pathway is just the movement of substances through the cell wall and intercellular space. The symplast pathway is through the cytoplasm, through the plasmodesmata. So they could ask it about the sucrose, they could ask it about water and movement of substances. It just depends in the question. Do you have any tips to stop feeling like scared, especially day of? I keep spiraling. Candy, the only thing I would say to you is know it's going to be challenging, accept it's going to be challenging. Ride the wave of adrenaline and cortisol that you're going to produce in the exam. Use that in a positive way. Acknowledge it'll be hard. Try your best. That's all anyone can do. Try your best. But by acknowledging that it's going to be hard, what you're going to actually do is help yourself to be able to get down on paper what you need to. The other thing I would say to you is when you come up against, and I do it all the time, I sometimes see a question in an exam paper, and especially if I'm in a class with students and I haven't done like hours and hours of planning, because I do a lot of planning for these, all these resources are mine. I spend a lot of time that you don't see. If I come up against a question and it makes me go, oh, and I panic, right? My students know this. If I panic at a question, I know that I'm panicking and therefore I am stopping my cerebrum from activating here. This is where I'll go into biology for you. You know that your conscious part of your brain, your cerebral cortex, is involved in that conscious part, that voluntary thoughts, okay? That thinking about things. When I am panicked, I am basically stopping getting up here. I get a blank screen. If I was in the car driving somewhere and I come up against a roadblock, am I just going to sit there for ages and wait and wait and wait? No, I'm going to find a diversion. I'm going to find another way to get me to that information. Now, that could mean that I go off in a tangent over here, but that will then slowly bring me back to the area where I need to be. So what I would suggest is if you panic, move on and come back, but do not leave any question blank. Even if you blurt down the biggest pile of rubbish that you think, you'll be surprised how many times my students have blurted on paper a load of rubbish, and actually there are some correct answers in there, so they can gain 1 or 2 marks from answering what they think is a load of rubbish. So that's the key thing I would say. I would say take your time when reading the questions, because you need to be sure that you're taking the time to understand what the question is asking you. Too many students skim read. Too many students see a keyword and go, oh, yes, and start writing an answer. And they've completely missed what that question's asking. So slow down, read it 3 times before you start answering. The first time you think you know what it's asked you. The second time you're more likely understanding what it's asked you, but by the third time you absolutely have got it. And if you've got a graph, or if you have a table of results, or if you have a table of an experimental design, dissect it. take the time, write notes to yourself in all different ways, because I do feel like that's where most students are going to lose their marking points. Oh, interesting. Eden, that is an interesting concept. Yeah, they could potentially. They could ask you about how there are some similarities and differences because it is about the movement of ions and co-transportation. So yeah. And that's what's nice because you're actually seeing them together. Yeah, they absolutely do. Honestly, if you are looking at Mariam, you are so true. They will put, it's not that they're they're using really bad graphs. They've designed them on purpose. They've designed them on purpose to make it obvious if you look, because that's assessing your module one content, your ability to evaluate and conclude from information that's been given to you. So, how many samples did they do? How many so what was their sample size? How many intervals did they assess at? Have they shown you error bars? Have they done any of that? That is all the bits that they're basically saying. If you've done the learning, if you understand it, you'll see it in front of you. And that's why it's important to know that. How would they link it though, unless they go on about, yeah, that is exactly what they could do. They could assess your knowledge on module two, biological molecules and transport across biological molecules, but do it in the situation of your plants transport and your kidneys. That's exactly what they could do because a lot of you go, oh, well, they're only going to ask me about respiration or excretion in the kidneys or this, but no, they're assessing you on the whole topic area. Modules 5 and module 6 is basically module 2 in context. So if you understand your module 2, whichever way they go, then you'll be able to gain marks by bringing that information forward. Right, I want to keep going and finish this one with the loading and unloading. So, excuse me, so you've got your sucrose and it will move into at the companion cell, move through into the cytoplasm, and then you're now into the symplast pathway. So apoplast pathway when it's going through the wall, symplast pathway when it's being loaded into the phloem subtubelement. So we know that xylem is transpirational stream. We know the water's being pulled up against the pull of gravity because of cohesion and adhesion. We know we're going to load our sucrose into the phloem subtube element. That's going to lower the water potential, pulling the water from surrounding cells. So remember our bordered pits allows for lateral flow and from surrounding cells. Therefore, creating a hydrostatic pressure, that's going to move to where there's a lower hydrostatic pressure, and that therefore means we can unload. What are we doing? We are unloading it at an area where it's either going to be converted to glucose and used in respiration, converted to a polysaccharide, amylose and amylopectin for storage in starch, or it's going to be converted into amino acids and allowed for the growth of the plants. So therefore, you've got the loss of the sucrose, increase in the water potential. It's going to go in the opposite direction. That is lowering the hydrostatic pressure, allowing for it to continue to go. Why can't glucose be transported? So the glucose, if the glucose was transported, it would therefore lower the water potential more. And so by doing it as sucrose, it doesn't affect the water potential as much because it's a disaccharide rather than a monosaccharide. Plus the glucose is the one that would be hydrolyzed down through the process of glycolysis. Amna, does that answer your question about the lowering of the hydrostatic pressure? Once you unload it at a sink, whether that's above or below, you are there for lowering the water potential. Yes, absolutely right. The Casparian strip is involved if we're talking about the root and we're talking about transport in roots when we get to the endodermis layer. So the Casparian strip is only involved in the apoplast pathway for moving it, the water movement into the Symplast pathway, and that's to prevent any substances that the plant doesn't need moving in because the biological membrane is selectively permeable. Yes, Amna, you're right. If the sucrose is used up, we've now increased the water potential. And that's why water will leave. Because remember, water moves from a high water potential to a lower water potential down the water potential gradient. So the water will move back out to surrounding tissue. And that's why it's going to move in that direction. Does that make sense? So you're getting confused, Amna. The water will only move from the xylem to the sieve tube when there is a lower water potential at the point where we have loaded. We're at the bottom, we're at the sink, so we're at the point where we're unloading. So wherever you are loading the sucrose, you are creating a lower water potential. So therefore, water will move in by osmosis. When you get to the point where you are unloading, you are raising the water potential and therefore water will move out by osmosis. Good. Okay. The last little bit then, just on this one, you're going up or down. It is bi-directional. Remember that. Your sink could be above, and if it's above, then we're talking about fruits, seeds, growing leaves. And one thing I do want to bring in is I do feel, again, I could be wrong on this, so don't quote me if I'm wrong on this, but I feel like they're potentially going to link this to ba 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 ba. Your fact that your sink is, for example, your fruits, your seeds, your growing leaves, but they could potentially become sources in storage organs, i.e., if we look at the seed and we look at germination, when water is absorbed, what happens is the hormone gibberellins are produced. They are going to be metabolically activated, and that therefore is going to switch on genes. Where were the genes? The genes were inside the cells in the nucleus. That's therefore going to have the proteins broken down into amino acids. We therefore have enzymes. Here we're talking about enzymes. Enzymes being produced. Well, amino acids are needed to build the enzymes. So remember, amino acids get added together through the process of translation. So protein synthesis. So we've got hydrolysis, but we've also got protein synthesis. And therefore, we've now got enzyme which can break down starch. Isn't this part also linked? Yes, exactly. This is what I'm trying to show you. We're talking about potentially transport in plants, also looking at the hormones in plants in the fact that that storage organ. So this is why I'm showing you that link. It's paper one. It's module one, module two, module three, and module five. They can go whichever way they want. They could even say, well, hold on a minute, our seed has starch in it. Could we do a quantitative test on the starch that's produced, the starch that's stored, and a quantitative test on the glucose that is being hydrolyzed into in this question? So it's an interesting one because I'm currently, the reason I'm saying that is I'm building my workbook about this because I have a feeling this question could actually, I'll give you a sneak peek. Let me bring it in. So my workbook on plant response, she says, so where's my workbook's gone? That's not where I wanted to go. I wanted to go to here. It's taken me so long to do these because I don't want to obviously rush anything. Yeah, absolutely. There's so much they can ask in so many different ways. And this is the thing that you have to understand that they're never going to go in just one place. They're testing your knowledge on it all. Right. Someone asked about practicals. I'm going to be nice to you and give you a sneak peek of my workbook that may help you on practicals for balancing the back. That's not the one I wanted though. Where's the one I want? Don't want that one, don't want that one. Don't want that one. I want this one. Okay, so digestion of starch to maltose, hydrolyze into glucose, aerobic respiration, or synthesis of cellulose. If they were to ask you about practical based on plant hormones and auxin, they could talk about the fact that you've got here no cap, you've got a translucent cap, bending of light that occurs. So you've got proving that the receptors have to be at the top. If you put an opaque cap, nothing happens. So therefore, that's a way in which you can prove the receptors are at the top. If you're looking at removing the tip, you're proving that the hormone auxin is produced at the tip and has to diffuse down. So remove the tip, no growth. Agar block with nothing, no growth. If I've got the diffusion and then I stick a block that doesn't allow diffusion to occur, no growth. Put it and offset in means that the auxin is at high concentrations on the shaded side, therefore bending occurs at that point. Or looking at what I was just talking about in the quantitative testing using germinating seeds. Imagine I've got an agar plate, and in that agar plate, I have a starch solution. What I could do is I could take some seeds, I could crush the seeds up, and I could take them and place the crushed seeds into a well. Now, if I put them into a well, we know if the seeds are germinating, they therefore must be producing amylase that breaks down starch. So if I put in my well distilled water, or if I put boiled germinating seeds, or if I put germinating seeds into the well, what happens after I leave them to incubate? Well, we know that if we use the iodine test, iodine is going to go blue-black if starch is present. So my whole agar plate should be blue-black. If I have distilled water, there's no enzymes, therefore there's no change. So enzymes are needed for the hydrolysis of the starch down. If I use boiled seeds, if I've boiled them, I've gone past the optimum. I've therefore denatured the enzymes. There's no starch present. If there's no starch present, if there's no starch being broken down, sorry, that therefore means that those enzymes were denatured, and therefore without the enzyme hydrolyzing, there's no breaking down. So I'm proving enzymes are required. And if I have my germinating seeds and I therefore leave them 48 hours, and now I can see an area, a zone, which is no longer blue-black in color, what I can therefore tell is that the starch that was present has been hydrolyzed down, indicating that the amylase is diffusing and breaking those starch stores down. So I could use that as a way of assessing it, or I could say, well, if I've got starch being broken down, it's broken down into my glucose molecules, I could do reducing sugar tests. If I'm looking at my sucrose, I would want to do a non-reducing sugar test on that one. So that would be the way in which you could bring in the information about qualitative and quantitative data. So you need to know about reducing sugar test, which is your Benedict's, and you need to know about your non-reducing sugar test, which is sucrose, as the example that you should know. So the sucrose has to be hydrolyzed using hydrochloric acid first, and then that has to be done with the Benedict's again, and you will then see your glucose and galactose becoming present. And then you've got your starch test, which is the iodine test, and you have your biuret test for proteins, and then you have your lipids test, which is your ethanol test. So qualitative No, Mariam, I won't do them on Thursday. I thought about this long and hard. I won't do it on Thursday, and the reason being, I think that it's better on the day to do it yourself without this like right now you can see that I'm like going blah, 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 blah. You've got my videos, do the topic areas, look at your flashcards, do whatever you need to do, but do it on your own or with someone you trust as a friend to go back and forth with for testing you. The reason being is you want to try and minimize your stress. People might start giving you wrong information. People might start slipping up on the day. You want to just get yourself into a zone of you're going to start, you're going to do your best, and you're going to try and pull that information forward. I will try and do, I will do a live on Tuesday and Wednesday evening, just as much as I always do, but it will be whatever I do at that time. The qualitative testing side of things would be you observing the color change. With the quantitative testing is about getting the numbers, and therefore, what I would say is on that front, you are very welcome. And good luck if I don't see you beforehand. And especially if you've got maths, because that's in. What was I saying? Quantitative numbers. So you'd be using the likes of a colorimeter for your reducing sugar tests, for example. Guys, I need to know, or you can actually measure the size of the clear zone. So you'll have done it when you did your microbiology one and you were looking at your inhibition zone of antibiotics and how they can kill the bacteria. So what I would say is measuring the zone by using a ruler could be another way you did this, for example. But again, it all depends what the question is. Guys, I am going to finish because I've got a meeting at 12:30 and I need to eat something. So thank you for those of you who have joined me. I hope you have found it useful. Obviously, do go check out my videos that are already on here on specific topic areas. The whole course is there for you. So you are very welcome. And yeah, thanks, guys.